Hello everyone, this is Medical Research Tools and welcome to another brain segmentation tutorial. In this episode, I am going to show you how to segment the whole cortex and white matter in the cerebrum. For start, I have to choose the imaging that has a clearer contrast and shows a better separation between cortex and white matter. So I go to data and check all the modalities that I have. And in this case, the T1 has a better contrast between cortex and white matter. Okay, in the segment editor, I add a new segment, and by using the threshold method, I try to grab the threshold that's best match with the white matter. Don't forget to choose the everywhere and override all in the segments below and try to be precise. And remember, in this episode, I am gonna change the whole process. For start, don't forget to press apply instead of use for masking. By pressing apply in this section, you put all your new segment on the selected threshold, which is in this case a white matter. Now I add another segment. It's better to rename your segments. The new segment is gonna be our cerebrum. So again, I use the threshold method and try to put the threshold on the whole cerebrum. I change the cursor in order to match the whole cerebrum. In this stage, it is important to change some of these settings to allow the overlap between these two segments. So I choose allow overlap, and in this stage, I press use for masking. Now we have two segments which in some parts they overlap. Now the pink segment, which is the representative of the whole cerebrum, I try to use the paint tool, and by checking the editable intensity range, draw some lines in the cerebrum area on multiple slices in all the different views of our imaging. Don't forget to check for the allow overlap in the settings. If you have watched the previous tutorials, you can find this method very easy and familiar. We put some seeds of our selected segment, and then by using the tool called grow from seeds, we grow these painted lines into our desired segment. But in this case, we have to choose the allow overlap to let the system overlap the pink over the previously selected green segments. So I repeat my work on multiple slices in different views. And then I have to add another segment, which is the representative of the all surroundings areas of the cerebrum. Now, uncheck the editable intensity range. Again, you have to choose the low overlap and draw some closed loops around the cerebrum. Repeat it on multiple slices in different views. The more you repeat your work, the result will be more precise and with less errors. Don't forget to separate the cerebellum. And make sure that your lines for the surrounding area is a closed loop. In this stage, I turn off the green segment or the white matter to better visualize my, in fact, cerebrum and the other segment. Don't forget to separate the cerebellum and brainstem because we are working on the cerebrum. Okay, after repeating the work on multiple slices and different views, now I go to Grow from Seeds tool and press Initialize. Again, don't forget to choose Allow Overlap. This tool will automatically segment the inner segment, which is the cerebrum, based on the boundaries and borders that we've drawn. Now, before finalizing the work, it needs some corrections. There are some leakages and some undersegmentations. Most of the times, there are some mistakes in the lateral ventricles. By using the paint tool and choosing the yellow segment, I draw a small line in the ventricle area and it will automatically correct it. And there are some mistakes around the occipital lobe that I try to check different slices in all views and correct the mistakes as much as it is possible. Don't forget to choose the right segment. For correcting the leakages, you have to draw a yellow line around the over-segmented area, and for some parts that is under-segmented, you have to choose the pink one. Don't forget to look over the skull as well, because sometimes there are some leakages on that part. Now go back to Grow from Seeds and press Apply. In this part, we have the cranium separated, which is overlapped on our white matter. So the next stage would be separating the white matter and the whole cranium or cerebrum that we have segmented. Uh, in this stage, I am going to introduce a new tool in the 3D slicer that lets us to subtract a segment with another. In this case, I am gonna subtract the overlapped parts of cerebrum with the white matter. Okay, I'll go to the logical operators tool. In this list, you have to choose subtract, uncheck the bypass masking, and choose again. Don't forget to choose overwrite all in this part. We don't need any overlap. And choose the segment that you want to be subtracted by another. On the top, we choose the cranium, and the bottom choose the white matter. In this case, the overlapped areas between these two segments would be subtract with white matter. By pressing apply, it results in what you're seeing. The white, we have two segments. One of them is white matter, and the other one is now the cortex. Also, there are some over segmentations around the skull because of the same opacity and contrast between white matter, between gray matter and the skull. In this case, again, by using the subtract tool, I subtract the over segmented blue parts with the yellow one. It is just like before. I choose the white matter at the top and subtract it with the yellow tool, which is the surrounding area, and press apply. Now we subtracted these mistaken segmented parts with the yellow segment. This is the results. We have cortex at the top, and then it is white matter. As always, our work needs some corrections. There are extra islands and extrusions. I use the island tool for erasing the extra islands. In this part, go to islands, press keep the largest islands, and press apply. It will delete all extra voxels and islands. And do this for the cortex too. And another tool for correcting the protrusions is the smoothing tool. It's simply press apply and it will smooth your structure in a symmetrical way. As you can see, there are some over segmentations of white matter and some leakage around the occipital area. And for correcting this part, I try to use the paint tool and draw the undersegmented cortex to fix this mistake. To make it easier, I use the threshold method and make sure to choose the threshold of the cortex, and by drawing around these undersegmented areas, I fix the mistakes. By using the threshold method and choosing the exact threshold, it will help you to not grow and draw out of you the lines of the cortex. Okay, after correcting and smoothing and deleting the extra islands, as you can see, there are some empty voxels in the segmented structure. For filling these empty voxels, there is a method. First of all, add a new segment, try to choose a very sharp color. And in this process, first of all, you need to fill these empty voxels with your new segment. I go to logical operators and choose fill. And in the bottom, I choose outside all visible segments. By pressing apply, the red segment will be put into all empty voxels, or actually outside all visible segments. Then I go to grow, to the grow tool, make sure to choose grow and minimize the size of growing or margin size, which I use 0.8. Don't forget to check apply to visible segments. And in the editable area, select the inside segment 4. Then press apply. It will go through all the segments and grows them into the segment 4 or the red segment in order to fill these voxels. Repeat it as much as it needs until there is no red segment. And simply, we have grown the adjacent segments into our empty voxels. This method is very helpful for fixing our some mistakes and some empty voxels into your structure when it is important to left no empty dots and voxels. As you can see, there's no empty voxels into the structure, and I can remove these red segments. And for the last part of our work, we have to fix all these extrusions of white matter through the cortex. Which in this case, I use the paint tool. For finding and localizing a specific region on your 3D structure on the 2D views, you can use the cursor method, cursor tool at the top right of the screen. When you check the cursor at the top right, and when you want to localize a region on all the views, you have to press the shift button and move your cursor around the selected area, and it will show you the exact region on all other views. In this case, you'll be able to localize the mistakes or the part you want to work on. And as you can see, I am drawing the undersegmented cortex areas. Another point worth to be mentioned is that there is a box called color smudge in the settings that I've checked. By checking this box, you'll be able to draw one segment to the adjacent area. For example, I draw my cursor from adjacent pink segment to the area that is undersegmented, and it will grow or draw this pink segment to the region that I want. And now I try to localize all the mistaken parts or undersegmented cortex parts in the 3D view in order to find all the errors and fix them, which apparently is done. And we have cortex and white matter segmented very clearly. Now, for adding some more beautiful details into your structure, I am gonna separate the left and right structures, which is simply doable with the scissors tool. I add another segment, call it left white matter. And by using the scissors tool, you draw a circle around the part that you want to change it to another segment. Now I subtract or change the blue part with the orange new segment, which is called left white matter. Don't forget to choose the desired and selected segment that you want to create in the settings below. And I repeat this for the cortex. Don't forget to rename your segments to not mix them up. And now I am gonna use the scissors tools to change the left part of the cortex into the new segment in green. Editable area should be the left cerebral cortex, and at the top you have to choose the pink one, which is cortex. Carefully, I draw my line around the left cerebral cortex, and it will automatically change into the left cerebral cortex in green. Now we have 4 segments very clearly separated in the brain imaging, left and right white matter and left and right cerebral cortex. Based on what you need, you can add more details and more segments into your structure. For example, you can separate all the lobes of the cortex, frontal, parietal, and occipital lobes, and add new segments as much as you want. Hope you found this tutorial very helpful. I suggest that you start trying to practice on 3D Slicer and try to actively use these tools that I'm showing in order to learn them and use them into your own future projects. See you soon.